A Silgard-coated cover slip, made by smearing a drop of Silgard across the glass and curing overnight, is placed in the circular cavity of the prefabricated recording chamber and covered with recording solution. A worm placed in the center is glued to the Silgard surface, starting from the head or tail, applying glue to the dorsal side of the worm using a glue pipette. Once gluing is complete, an incision adjacent to the vulva at the interface between the cuticle and glue is made with a glass needle and extended towards the head. Following extraction of viscera and eggs from the body cavity, the cut edge of the incision is spot welded with the glue to the Silgard, exposing the ventral nerve and body wall muscles. The ventral medial muscle anterior to the vulva is patch clamped, and the stimulating electrode is placed upstream on the nerve cord.
Hello, I'm Janet Richmond, an Associate Professor in the Department of Biological Sciences at the University of Illinois at Chicago. Today I'm going to show you a dissection technique which allows us to expose the neuromuscular junctions of C. elegans for electrophysiological recording. We use this prep to study synaptic transmission. Okay, let's get started.
Before working with the nematodes, some hardware must be crafted at least one day before the procedure. A dissection recording chamber, Silgard-coated glass cover slips, pulled pipettes, a glue applicator, and an extraction pipette. Begin by constructing